Hello, doctors of medicine here. Today we bring you the first video of our histology playlist, 100% beginner-friendly introduction to histology. Let's get right into the video. So these are the contents of our video. There's definition of histology, microscopy, tissue prep, dyes and staining, and then a tiny conclusion. What is histology? This is probably the question that is in everybody's minds right now. So histology is the study of body tissues and how they make up organs, all right? How they are arranged to make up organs. Okay, that is histology, the study of body tissues and how they are arranged to make up organs. So when we say tissues, we are referring to a group of similar cells that are arranged together to perform a certain function. Let me expagorate further. Let's take a look at the picture over here. We have a cell. This is one cell that I'm highlighting over here. This is one cell. And this is a group of cells over here. This is a group of cells. And these cells, they are similar. So we call this group of cells a tissue. But mind you, a tissue is not only formed by a group of similar cells. It is also formed by something that we call extracellular matrix. This extracellular matrix is basically just a substance that is made up of different molecules, and its function is to support these cells. Okay, right. So that is the definition of a tissue. Now, let's start from the top. I want to give you an idea of what to think whenever the word histology pops up in your mind. Let's start from the top. We begin with a cell over here. This is a cell. Okay. So the cells, they make up a tissue. Okay. This is a tissue that is made up of cells and extracellular matrix. These are the definitions of a cell, definition of a tissue, the definition of an organ. So the tissues, they then make up an organ over here. Right. Now that is histology. The study of these body tissues, including how they're formed, how they function, and even how they respond to certain conditions such as infection by a pathogen, and most importantly, how they make up organs. The study of body tissues. Now, let's delve a little bit deeper into our tissues. We have four basic tissues that we're going to focus on in histology as a subject. Number one, there's nerve tissue. This is nerve tissue over here. Number two, there's connective tissue. This is connective tissue. Did you know that blood is a form of connective tissue? Well, now you know it. Number three, we have epithelium. Epithelium. Number four, we have muscle. Right, so these are the four basic tissues that we're going to focus on in histology, but we'll make separate videos for each tissue. Right now, just know that there are four basic histological tissues. Now, let's talk about how exactly we study these tissues in histology, the how part. Let's talk about microscopy. In histology, we study organs and their function from cellular level. So we use microscopes to view the specimen because we cannot see cells with our naked eye. Okay, there are two types of microscopes that we learn about at the University of Zimbabwe. Well, there's a light microscope and an electron microscope. So you need to be able to note the differences between these two microscopes. I just listed two differences over here. So the maximum resolution of a light microscope is 0.2 micrometers, allowing a magnification of 1000 to 1500 times. Whereas the maximum resolution of an electron microscope is 3 nanometers, which, permits a magnification of 120,000 times. Okay, so before I move on, let's do a quick definition of terms. Resolution, the resolution that I was talking about over here, Resolution is defined as the extent to which a microscope can distinguish between cell organelles. So when we said the resolution of an electron microscope is 3 nanometers, we meant that the electron microscope is going to view organelles that are larger than 3 nanometers. That's the first aspect of resolution. The microscope can view organelles which are only larger than 3 nanometers, the electron microscope. And the second aspect of resolution is that the microscope, it can distinguish between adjacent organelles that have a distance of greater than 3 nanometers between them. Okay, let me use an example over here. In a cell, you have 2 mitochondria. This is one mitochondria, and this is the second mitochondria over here. So these mitochondria, they're separated by a distance of less than 3 nanometers. Okay, so the distance over here is less than 3 nanometers. This means that, okay, let me just write the nanometers over here. Yeah, this is taking so slow. All right, so we have 2 mitochondria over here. This is one mitochondria, and we have the second mitochondria. 2 mitochondria, and between them is a distance of less than 3 nanometers. What is your microscope going to view? Your electron microscope, it's going to view these 2 mitochondria as one, because the distance that is between these 2 mitochondria is less than 3 nanometers. It is too tiny. Right, it surpasses the resolving power of the microscope. So your microscope is going to view them as one. But if the distance between these 2 adjacent mitochondria was greater than 3 nanometers, your microscope would be able to distinguish between the 2 mitochondria. You would be actually able to see that they are 2 mitochondria instead of 1. All right, that is the definition of resolution. So over here, there's just a little table that shows measurements. 1 centimeter is equal to 10 millimeters and stuff. So you might need to write it down if you didn't know it already. Let's move on to tissue preparation for study under a light microscope. So we're basically going to be describing how exactly we prepare our tissues for study under a microscope. All right, so we have a sample. Over here, we've taken a sample from a donor. We have to prepare that sample so that we may be able to view it clearly under a microscope. So the first step in tissue preparation is fixation. Fixation involves the use of a chemical that is known as a fixative to inactivate degradative enzymes that may be released by the cell. Hence, fixation is done to preserve tissue structure. The second step over here is dehydration. So dehydration is basically the removal of water, and this is done by alcohol. So the tissue specimen is immersed in different concentrations of alcohol in order to remove water. The third step is clearing, and this basically means that we're removing the alcohol that we had used in the second step. So we're clearing this tissue specimen of the alcohol. The fourth step is infiltration. So the tissue is infiltrated with molten paraffin. It is placed in molten paraffin so that the paraffin can infiltrate into the specimen. And then the fifth step is embedding. Embedding involves pouring the infiltrate into a mold, allowing the paraffin to harden. And then the sixth and final step is trimming, which is the sectioning of the paraffin block by a microtome, which sections it into very thin slices to allow light to pass through the specimen so that we may actually view the specimen under a light microscope. So those are the steps that are taken in tissue preparation for study. Pause and review if necessary. So after trimming, the sample is not yet ready for study. You literally will not see anything. So there's need for staining, and this is done by the use of dyes. Now let's talk about dyes and staining. So the dyes used in histology, they selectively stain cellular organelles. They do selective staining of cellular organelles. How is that, you may ask? Well, the dyes often behave like acidic or basic compounds, and they form electrostatic linkages with ionizable radicals of macromolecules and tissues. Okay, that was a lot of jargon, but don't worry, I'll explain everything in full. Let me expagorate on how exactly dyes do selective staining. The most common type of dye that we use in histology is hematoxylin and eosin. All right. It's a combination of these two chemicals, and it can also be called an H and E stain. H and E stain. Right. Hematoxylin is basic, so it will stain cellular components with acidic residues in them, and it stains these cellular components blue or purple. Let me explain further. Oh, before I explain anything further, let me talk about eosin. Eosin is acidic, hence it will stain basic cellular components, and it will stain them pink or red. Cellular organelles can be acidophilic or basophilic over here. So when we say philic, we mean loving. For example, hydrophilic means water loving, so acidophilic means acid loving. Basophilic means base loving. So for something to love an acid, that is, it being acidophilic, it has to have basic residues in it, right? Because acids react with bases, right? So I was saying that hematoxylin is a basic dye. So it being basic, it will stain basophilic cellular components, that is, cellular components that love bases. That is, cellular components that have acids in their organization. Which are these cellular organelles that are stained by hematoxylin? Well, we have DNA, we have RNA, and we have glycans. Eosin, on the other hand, is an acidic dye. So it stains acidophilic cell components, that is, cell organelles that have basic residues. Which ones are these? These are mitochondria, secretory granules, and collagen. So basically anything with a lot of protein molecules can be stained by eosin. So this is a small table that shows other types of dyes besides hematoxylin, and yeah. So under acidic dyes we have eosin, we have orange G and acid phosgene. Under basic dyes we have toluidine blue, Alcian blue, and methylene blue. That brings us to the end of this video. In the next video, we're going to be studying the cell and cellular organelles for the sake of those who are not so well versed in biology. If you have any questions, please do write them up in the comments. And if you enjoyed this video and it was helpful to you, please do give us a thumbs up and subscribe for more content. Best of luck in your journey. Cheers.